Chapter 10 in the OpenStax book talks about biotechnology. Biotechnology, by definition, is the use of artificial methods to modify genetic material of living organisms or cells to produce novel compounds or perform new functions. Very fancy definition. One of the hallmark pieces of equipment is this little fellow right here on the left. That's a thermocycler. It's a really basic tool used to study DNA through a process called polymerase chain reactions. The polymerase enzyme most often used with PCR comes from a strain of bacteria that lives in a hot spring of Yellowstone National Park. I really like this juxtaposition of something really old next to something that's relatively new, because biology, while trending towards these really cool pieces of technology, still kind of relies on the old, which I just love. So anyway, let's talk a little bit about biotechnology. To manipulate genetic material, first we must be able to extract it. While there are simplistic ways available to anyone with a kitchen, there are more kind of precise methods that scientists prefer to use, although a lot of the steps sound the same. Scientists will first lyse cells or make them burst by using detergent, and that gets the plasma membrane out of the way and essentially turns it into a bit of waste. Cell contents are treated with proteases, or enzymes that break down protein to get the proteins out of the way. They are then treated with enzymes that destroy RNAs to get the enzymes out of the way. That whole now mostly destroyed kind of cellular muck is put in a machine called a centrifuge, which spins it really, really fast. And a lot of those solid materials head to the bottom of the tube. We call them the pellet, and then the liquid on top, which is called the supernatant, is still going to contain your DNA, and it can be transferred into a nice clean test tube. The DNA is precipitated or made to fall out of solution when we add ethanol, and it turns it into like a viscous, quite frankly, snot-feeling substance that you can wrap around a glass rod in order to manipulate later in some other fashion. Once we have isolated DNA, there are a lot of things we can do with it. Running it through a gel to look at the lengths of well-known hallmark sequences can teach us quite a bit. This process goes back to our concepts of size and diffusion rates. Smaller bands or pieces of DNA can travel further in the gel, while larger chunks don't move as quickly. We end up with characteristic banding patterns like you see here. Repeats in certain sequences of both entire species and unique individuals have been mapped. Therefore, we can identify an unknown bacteria by looking at its banding patterns in gel electrophoresis, or we could match a crime suspect to a list of individuals we believe may have been involved by matching their banding patterns. It's not as simple, though, as just taking a single cell, removing its DNA, and running it through a gel. Often we have to amplify it or make many copies, so we have enough to see in those bands first off, but then we want to make sure that we have enough material to accurately test it. The copying process we often employ is that polymerase chain reaction mentioned at the beginning. In PCR, a single piece of DNA is denatured or split in two by heating it to 95 degrees Celsius. Then we add a pre-made primer in a process called annealing after we cool the sample to negative 50 degrees Celsius. DNA polymerase extends the fragments in a warm 72-degree environment, and the new pieces head back up to that denaturing step. So we made 2 new pieces of DNA from one little double strand, so the next time around we'll make 4 and then 16, and the growth of perfect copies is therefore exponential. This is how we can prepare small samples for larger tests relatively quickly in the lab. Molecular cloning is another neat outcome of biotechnology. In molecular cloning, multiple copies of genes, mRNAs, or gene products can be made and studied. This is easiest to do and see in bacterial vectors, and there are numerous uses of this process. Some can be found in the medical field, others in things like soaps and cleaners, which are often enzyme-based, and even fashion, where a particular enzyme makes stone-washed jeans. So let's look at an example of molecular cloning. Here you see a piece of foreign DNA and a very specific plasmid that has a LAC-Z gene and an ampicillin-resistant gene. We're going to use a restriction enzyme to cut the LAC-Z gene in half and to also cut out a piece of foreign DNA. The restrictive enzyme is going to cut those two individual sections the exact same way and leave them with sticky ends that match. Those sticky ends can be stuck back together using DNA ligase, which ensures a strong DNA backbone. The plasmids, which originally came from other bacterial cells, will be introduced into a new bacterial culture. The bacteria that uptake this plasmid will now be resistant to ampicillin, which is an antibiotic, and they will also not have a functioning LAC-Z gene because that's where we interrupted it with a piece of foreign DNA. When allowed to grow in culture, individual cultures of ampicillin bacteria will grow. Some of the cultures are going to be white because they're missing the LAC-Z gene. It normally turns the cultures blue, as seen in the picture. We then know that we interrupted the LAC-Z gene with a piece of foreign DNA and were able to stick a functioning plasmid back into a piece of bacteria. If you're a little fuzzy on this concept of a restriction site, you can see it here. In A, there is a regular 5' to 3' piece of bacteria. In B, you can see where the enzyme cuts, and in C, you can see the resultant sticky ends. If you have those sticky ends from two different organisms, there isn't anything that states that the original pieces of DNA are what need to be put back together. This allows us to create those recombinant plasmids, or plasmids that have been recombined from two different organisms. Organismal cloning is another outcome of biotechnology. In this case, a nucleus is removed from a donor egg. The enucleated egg is placed next to another cell, which was donated from a different individual, and they are shocked with electricity, which causes them to fuse. This injects the donor nucleus into the donor egg. The cell is then shocked again, so the cells must start division. Cells are allowed to divide for several days until an early embryonic stage is reached. This early embryonic stage, known as a blastocyst, is implanted in a surrogate mother, and she's allowed to undergo pregnancy as per usual. This implantation process is similar to what you hear about in traditional IVF. Only in this case, the resulting baby that's born is an exact clone from your donor nucleus organism. There are now a few different ways to do this experiment. You can simply remove the second nucleus and insert it into the egg, but the result is the same, a cloned individual. Now we're simply going to run through a couple of different things that we can do using biotechnology. This diagram shows you the steps involved in curing a disease with gene therapy using an adenovirus vector. In this case, we inserted a gene that an individual was missing into a virus. Viruses do an excellent job of inserting themselves into their host DNA. We're going to use the adenovirus to insert a healthy gene into a host cell. Hopefully, this individual will be able to use the gene products made by the insertion of this brand new gene, and it will correct some sort of protein deficiency that they have. Here we can see another example of biotechnology in the form of transgenic mice. They have genes that cause them to fluoresce under a UV light. The non-transgenic mouse in the middle does not have that gene that causes fluorescence. If you're curious where a gene is active but you're not quite sure, you can put a UV gene next to your gene of interest. Anywhere the UV gene glows shows you where your gene of interest has also been made. Another common example of biotechnology use are the creation of GMO crops. GMO crops can create foods that are less resistant to drought, some that better handle storage, and others make foods that create more nutrients. GMO crops have allowed us to feed millions more individuals. Unfortunately, some people don't understand basic science and don't know that all of their food contains DNA. There isn't anything dangerous about eating GMO foods. It works exactly like any other food that you eat. Your body absorbs the nutrients. It doesn't take DNA from things that you eat and use it to modify your own genome. We spend way too much time protecting our genes to add random pieces. Biotechnology has also allowed us to create physical maps of particular chromosomes. In this case, you can see a physical map of the human X chromosome. And you can also see to the right-hand side different diseases that we've been able to find on these specific locations. Now that we know where these genes are, we might be able to modify that particular section of a chromosome. Maybe we can repair it, or maybe we can replace some of the missing gene products. We've been creating these genetic maps for entire organisms, not just singular chromosomes. Here you can see some of the model organisms that we've been using in scientific experiments for years. Saccharomyces cerevisiae is probably one of the most common yeasts that will ever exist. Thanks to the biotechnological study of Saccharomyces cerevisiae, we now know its entire genome nucleotide for nucleotide. It allows us to essentially reassess some of the information that we found in a broader but more in-depth way. Sometimes we find the DNA sequences of multiple species within a single environmental niche. This is called metagenomics. In this case, DNA is cut up and sequenced from many different genomes all at the same time. The sequences are reconstructed thanks to their overlapping pieces, and we can compare different genes from different species all in one fell swoop. This can be really helpful if we're curious about the environmental effects of something like a pollution or increased radiation or heat. We can sequence DNA, introduce these external factors, and then sequence it again and see if the DNA was damaged or changed in any way. Another neat use of altering the DNA of specific species like algae is the creation of renewable fuels. Turns out that some algae actually create lipids that can be used to power vehicles. If we could increase that lipid production or make even better lipids, we might be able to use our own fuels instead of having to drill into the planet and destroy entire ecosystems to get oil. On a more serious note, part of biotechnology is studying certain organisms that could cause mass disease or be weaponized if something ever went wrong. An excellent example is Bacillus anthracis. This is the organism that causes anthrax. In some places, biotechnology is being used to attempt to weaponize anthrax and make large amounts of people sick. This could lead to bioterrorism or biowarfare. Thankfully, there are far more scientists who are looking at ways that you would stop one of these attacks or treat weaponized genetic material in mass. On a happier note, there are some transgenic agricultural plants that are being made resistant to disease or attacks. This will help us to protect our food chain. As the human population explodes in numbers, it's very important that we can treat different plants or even animals for genetic illnesses and make them resistant, so they are less likely to die off due to infection. On one final note, we take a look at a very interesting piece of technology that's preparing to do a proteomic pattern analysis to identify specific cancers so that an accurate cancer prognosis can be made. What this is stating in a long-winded way is that some cancers have too much of one type of protein or too little of another. We can use that protein signature to diagnose or prognose cancer. We can also use that signature to treat the cancer. Are you missing a specific protein? Maybe we can give it to you and help you battle the effects of your cancer. Do you have too much of another kind of protein or enzyme? Maybe we can give you a medication to decrease the naturally occurring amount to get you more in baseline of what's seen as an average. giving us more time to treat the actual cause of the cancer rather than just treating its effects. With that, we end our chapter, and I'm going to strongly encourage you to find some videos. A lot of this biotechnology is really cool and very visual. You should go online and search for a topic that you find interesting. If you explore a single topic that you love, it'll open your eyes to the whole field of biotechnology. Be sure, of course, to read your book, taking notes in your own words prior to attempting your homework assignment.